Welcome to our second podcast on the urinary system, and in this podcast we will be looking at the physiology that's involved in the production of an ultrafiltrate from the blood. But before we actually dive into the different mechanisms and the regulation of this, let's watch a video that will basically review some of the things we learned last time and introduce us to the topic we're moving toward in this unit, the movement of water by actively moving sodium.
The kidney is the body's filtration system and produces urine in its thousands of nephrons, the kidney's basic unit. Each nephron begins with a Bowman's capsule wrapped around a capillary ball, the glomerulus, which becomes a tubule that eventually forms the loop of Henle, before collecting into tubules and ducts that empty into the bladder. Fluid from the blood enters the nephron by filtration and passes along it, where substances are added from the blood by secretion or removed from it by reabsorption. Potassium and hydrogen ions, organic acids and bases, metabolites and foreign substances are secreted across a selective membrane. The potassium and hydrogen are exchanged for sodium along the tubule. 99% of the filtrate is reabsorbed through a mixture of passive diffusion down an osmotic or concentration gradient and active transport by pumping against a concentration gradient. The kidneys can conserve water. The only way that the body can move water is by moving sodium chloride first. You have to create a concentration gradient so that water can move by osmosis in the direction of the highest concentration.
Now, as we look at the forces in the kidney that are going to be driving this filtrate from the bloodstream into Bowman's space, we see it's the same two forces that were at work driving the fluids out of our capillaries and into our interstitial spaces. And largely that's the same thing. Instead of interstitial space, now we're in Bowman's capsule. So we have our hydrostatic pressure, we have our colloid or osmotic pressure, and just like we saw with our Starling forces, we can calculate that movement as well. But the hydrostatic force is clearly going to be forcing material out of the blood and into Bowman's space. Therefore, this process is referred to as filtration. So our net filtration pressure, remember it's the difference between Bowman's capsule pressure and the capsule in the blood vessels, we see it's only about 10 millimeters of mercury. But that is plenty sufficient for what we're going to need to produce. And as we get to our third unit and we really talk about the physiological measures of the urinary system, we're going to talk about something called glomerular filtration rate. So if we take and look at the production of urine with that 10 millimeters of mercury, that production is going to be about 120 milliliters per minute. That's our glomerular filtration rate. We're going to see that equals about 7.5 liters per hour, or 180 liters per day. So essentially, by producing this much filtrate every day, when you look at the average rate that's moved, your entire blood volume is going to be filtered every 40 minutes. That's a highly efficient filter. Now, the way this is principally going to happen is in the different parts of our labyrinths. We see that we are going to form our filtrate in Bowman's capsule. We're going to modify it through our proximal tubule, loop of Henle, distal convoluted tubule, and later we'll see we're going to really change the water content in our collecting duct. Materials are going to move out of the ultrafiltrate into the tissues and back into the bloodstream. And do you remember what we called that when we talked about capillaries? Filtration was moving out. What did we call moving back in? Absorption. So we'll be talking about absorption as well in that modification. And in the third unit, we'll also see that we can add back to the filtrate down in these other portions, and we're going to talk about secretion. That's one we're going to add to it. But for now, let's look at what's happening here with the production of ultrafiltrate, changing the osmotic pressure, changing the hydrostatic pressure. We see that we're going to have some regulation, and certainly sympathetic activity can do that. If we cause constriction of either the afferent or the efferent arterioles more or less, that's going to impact the hydrostatic pressure on the glomerulus itself. Now, we also have autoregulation that's rather intrinsic. And so myogenic intrinsic regulation, the smooth muscles of the afferent arterioles, they can sense and adapt to pressure. So if the blood pressure is too high, then these afferent arterioles are going to constrict to resist the flow of blood into the glomerulus and prevent the pressure getting too high. Because remember, these capillary beds are only one endothelial cell layer thick. They're very not sensitive, fragile, if you will. And they have a lot of pores in them, the fenestration. So if the blood pressure gets too high, it would blow the glomerulus apart. So this is a protective mechanism. But we also have a means of protecting against hydrostatic pressure and monitoring it as well, and it is called tubuloglomerular feedback. And this happens in this juxtaglomerular apparatus region. We talked about that a little bit when we talked about hemodynamics. What we're going to see in the portion of the distal convoluted tubule that comes back in its direction back up into the cortex, it's going to pass immediately beside the vascular pole of its own renal corpuscle. And when it does that, it's going to be able to sense pressure differences and affect the smooth muscles of the afferent arteriole. So what am I talking about? Here we see a renal corpuscle. We see our loops of the glomerulus. We see our podocytes on the outside. Bowman's space, proximal convoluted tubule. Off the screen is the rest of the proximal, the loop of Henle, but the distal convoluted tubule is going to return and pass just in this region. This is our juxtaglomerular apparatus, and here we see the cells of the distal convoluted tubule in this region very closely opposed in here. They're so tightly packed it looks like a dense spot, which is what macula densa literally translated means. So these cells are going to be able to sense these pressure differences, affect the smooth muscle cells in the afferent arteriole, and therefore adjust the blood pressure of the blood as it's entering the glomerulus. That's some levels of regulation that we see. Now let's get to working with this ultrafiltrate. We've produced 180 liters a day, but we're going to modify it so that we only have about 1.5 liters that we actually excrete in a 24-hour period. Now, we have to produce a certain amount, 40 milliliters, for instance. This is the minimal volume we need to remove the metabolic waste, principally the nitrogenous waste, from protein degradation and metabolism. And so this obligatory water loss is going to amount to 400 milliliters a day. All right, so no matter how dehydrated you are, you've got to produce that much urine. So as we look at modifying this and that resorption of water, like we saw in the video, that's going to happen by osmosis, by creating that osmotic gradient. So we move solutes out of the filtrate into the interstitium and into the blood vessels of the interstitium, and that's going to be sodium. Sodium is going to be transported out. Chloride is going to follow. Now you have the salt, which is a solute, creating a hypertonic environment. Water is going to passively follow by osmosis. Now, what we're going to see in the proximal convoluted tubule, the very beginning part and even the descending thick part of the loop of Henle, 85% of the ultrafiltrate, the volume, is going to be reabsorbed. This is unregulated, it's going to be constant, and there's nothing you can do. You can't change it. That's going to happen because of this movement of sodium and because of the passive following of chloride and the osmosis of water. So in the proximal convoluted tubule, when we look at what's happening, all of the solutes that are present in this ultrafiltrate, they just simply flow through those 3 filters, remember. We've got the endothelial fenestrations, the pores, the basal lamina, and the slits of the podocytes. So all these solutes come through, so the ultrafiltrate in the beginning of the proximal convoluted tubule is isoosmotic. So it's 300 milliosmol. But once in the proximal tubule, as we saw in the video and as we see here, sodium is pumped out. Sodium-potassium pumps, they're our old friends. As they're pumped out, chloride follows. You create that hypertonic environment, and the proximal convoluted tubule is permeable to water. Therefore, water can move passively. That's an important part. Wherever we talk about permeabilities in these different portions of the loops, you need to make special note of those. So here, the proximal convoluted tubule is permeable to water. So we add a little bit to our illustration. Here we're looking at a renal corpuscle. We see the filtration, the production of this ultrafiltrate, materials moving out of the bloodstream into Bowman's space. But once we get into the proximal convoluted tubule, active transport of sodium, chloride, because of its ionic difference, is going to follow. So all of these are going to be solutes, and since these are ions, sodium chloride, remember, has two. It doubles the osmotic pressure because it dissociates, and now water follows by osmosis. This is resorption in blue, filtration here in red. Now, water is going to be removed, as is salt. Salt's pumped out, water follows. So really, as we move down the proximal convoluted tubule, we're removing equal amounts. And so we're maintaining this 300 milliosmolarity of the ultrafiltrate. So it's still isoosmotic. By the time we finish this, 65% of the salt and water have been resorbed in the proximal convoluted tubule. Now, as we start down the descending limb, we have that thick portion. We're going to remove another 20%. And again, this is unregulated. There's nothing you can do about it. And by the time we get into our loop of Henle, we're going to have about 27 liters remaining. And that we can regulate, and we have that much to play with as far as being able to conserve or excrete more or less of that fluid. Now, this is probably one of the most challenging parts of physiology of the kidney to understand, and this is going to be occurring in the loop of Henle, and this is called the countercurrent multiplier system. Basically, we're going to set up a cycle where we are moving sodium and water is going to move passively. However, we are going to be doing this in very specific locations. One side of the loop is pumping the sodium and is not permeable to water, and the other side of the loop is not pumping sodium, but it is permeable to water. And because of the direction of flow of the filtrate, because of the direction of movement of the sodium and chloride and then the water, you're basically going to create a cycle or a wheel that's going to continue to move and therefore cause water to be absorbed out of the filtrate into the interstitium and then make its way into the blood supply again. So that's where we're going. We're painting you a picture of that cycle. And it's called the countercurrent multiplier. So again, we're working with sodium and chloride. That's what we're going to move. Sodium is going to be actively transported. All right, again, it's a sodium-potassium pump. And this is going to be happening in the ascending loop of Henle, the ascending limb. So we dive down into the medulla, and as we're coming back up, that ascending limb is going to actively pump out sodium. Because we're pumping out sodium, chloride is going to passively follow. But look, down here at the bottom, the ascending limb is not permeable to water. And so as we pump out the salt, keep the water in, notice our filtrate's going to become hypotonic, maybe down to 100 milliosmol. But what's happening to the interstitium? It's getting increasingly more hypertonic. So what I want you to remember about the ascending limb, we actively transport sodium, chloride follows, water cannot. So here we have our ascending limb. This is our descending limb of the loop of Henle. We haven't covered it yet. Here we see our sodium pumps. Now, I really don't like how they've illustrated this. They've got it explained here. Active transport of sodium. These are our sodium-potassium pumps, but chloride is going to passively follow. This is going to create a hypertonic environment outside of the loop of Henle, down in the medulla. But notice as we move up, as more and more sodium chloride is removed, our filtrate's becoming more and more hypotonic, below 300. So that's the ascending limb. Now, let's look at the descending limb. Since we have pumped all of that sodium chloride into the interstitium and created a hypertonic environment, the descending limb is permeable to water, but it's not permeable to sodium chloride. So now water is going to be attracted to that hypertonic environment. Water is going to leave the descending limb by osmosis. And as water leaves, the filtrate is going to become more and more hypertonic itself. And actually, the deeper you go in the medulla, you're going to see that the filtrate becomes as hypertonic as the environment. And as you ascend back up into the cortex, the filtrate becomes it decreases. And so it's going to become isotonic and then hypotonic. So when we put this together, here we can see the descending limb, water is being removed, and we've got sodium chloride all over here. It's not that water is running away from the sodium chloride. But as we come down, starting with isotonic, as more and more water is removed, see what's happening? We are getting more and more hypertonic. The deepest part is going to be the most hypertonic environment. And here at the base of the loop, our filtrate is going to be 1400 milliosmol, and our interstitium is going to be 1400 milliosmol. These will equilibrate. But as we begin to ascend, we begin to pump out sodium chloride, but water can't follow. And so it's going to return to isotonic and possibly even go a little hypotonic as we remove more and more of the salt and keep the water. So the salt comes out, that drives the water out, but we do not want this hypertonic solution to possibly go back into the blood. We're going to return it to isotonic before we move into the collecting tubule and down our urinary tract. That's the countercurrent multiplier system. Now, to try to put this together just a little bit more, here is a short video that talks about this movement and puts it into motion a little bit. So this static two-dimensional image really doesn't give you a deep appreciation for what we're doing. Here's an animation that hopefully will help bring this together.
There is no initial osmolarity gradient in the kidney's interstitial fluid, where the osmolarity is identical to that of normal body fluids. The first step involves sodium leaving the ascending limb, either by passive diffusion in the thin ascending limb or by the NAK2Cl cotransporter in the thick ascending limb. Because the thick ascending limb is impermeable to water, water stays behind and the osmolarity of the thick ascending fluid decreases. Sodium is added to the interstitial fluid, increasing its osmolarity. This creates an osmolarity difference between the interstitial fluid and the thick ascending limb. Water diffuses out of the thin descending limb until the fluid there has equilibrated with the interstitial fluid. Once the descending limb has equilibrated, fluid from the proximal tubule enters, pushing the higher osmolarity fluid below it down the loop of Henle. The osmolarities of the fluids in the descending limb and the interstitium are again mismatched, causing water to leave the descending limb until the two fluids have re-equilibrated. You can see a corticopapillary gradient being generated between the fluid in the cortex and the fluid below. These two steps are repeated with each cycle enhancing the gradient. Sodium leaves the ascending limb for the interstitial fluid, decreasing osmolarity in the ascending limb. The interstitial fluid osmolarity increases. Water diffuses out of the thin descending limb, increasing the osmolarity in the descending limb until it equilibrates with the interstitial fluid. Low osmolarity fluid from the proximal tubule pushes the fluid below it down the loop of Henle. Water leaves the descending limb to decrease interstitial fluid osmolarity and re-equilibrate. At its maximum, the osmolarity of the inner medulla is 4 times that of the cortex.
So now that we have an understanding of the countercurrent multiplier, the dealing with the ultrafiltrate in the loop of Henle, we've gotten the sodium and chloride, we've gotten that into the interstitium, we've gotten the water into the interstitium, but now what do we do with it? How do we get it into the bloodstream? And this is where the vasa rectae, remember the arteria rectae and the vena rectae, how they then accumulate this material and remove it from the kidney and thus effectively reabsorb it back into the circulatory system. So we're going to see the descending limb, the arteria rectae. This is going to have molecules that are called aquaporins. These are channels that allow for the diffusion of water through these hydrophilic passageways. We're also going to have urea transporters, and we'll talk about the function of urea in just a few slides. But on the ascending limb, the vena rectae, these are also going to be fenestrated for rapid diffusion. So we're going to diffuse materials in or out of our vena rectae and therefore remove it and modify it from the interstitium of the kidney. Now, just to review, here's the descending limb, the arteria rectae. Here's the ascending limb, the vena rectae. And this is what we're going to be dealing with because we're surrounding that loop of Henle. So with our vena rectae, we have a countercurrent exchange, not to be confused with the countercurrent multiplier. All right, so don't do that. So here in this countercurrent exchanger, we see in the descending limb, sodium chloride, urea, they're going to enter the circulatory system, and very little water is going to leave at this point because the sodium and chloride, we're increasing that osmotic gradient. But in the ascending limb, sodium chloride, urea, they're going to exit. They can diffuse back out, but water, vast volumes of water are going to enter back into the vasa rectae and leave the kidney. So what's going to happen? This recycling of the solutes, removing the water but putting the sodium chloride and urea back in, it's going to maintain a hypertonic environment, but the net movement of water is out of the filtrate into the hypertonic interstitium, into the vasa rectae to maintain the osmotic balance and the osmotic pressure of the plasma itself. So as we see in our illustration, as the descending limb moves down into the medulla, remember this is increasingly hypertonic environment, we're going to see the salt goes in. The salt's following that gradient. Now, we cannot put this level of salt back into the bloodstream. We can't do it. So in the ascending limb, as this moves up, the solutes, the sodium, the urea, they exit, and a lot of water comes in so that by the time our ascending limb gets ready to leave the kidney, it's back to 300 milliosmol. It's back to that osmotic balance and therefore safe to return to the body. Now, you may ask, if we've got such a hypertonic environment in the interstitium, then why in the world would water go back into the bloodstream? Well, remember, it's hypertonic based on salt. And even though we're looking at this hypertonic environment, it's extremely hypertonic compared to our ultrafiltrate. But when we look at the proteins that are present in the plasma, that oncotic pressure is going to be a tremendous force then to draw the water out of the interstitium and into the vasa rectae. Now, we mentioned urea just a little while ago. Urea is going to be, again, a byproduct of nitrogenous waste, but we see that it's also going to function as a solute in the interstitium. And we're going to have a recycling of urea, much like we've seen with sodium and with water in our countercurrents and with our vasa rectae. But here what we're going to see is that both the ascending limb of the loop of Henle and collecting tubules are permeable to urea. So they're going to be able to diffuse. But we see that collecting tubules are going to contain urea transporters. And these transporters are going to transport urea out of the ultrafiltrate and into the interstitium. So basically what we're going to do, we're pumping out urea from the collecting tubule area. It's going to diffuse into the interstitium, but it can re-enter by diffusion into the ascending limb of the loop of Henle, and therefore we're going to see another cycle of recirculation that's going to happen. So here we have our descending limb. This is going to be our proximal convoluted tubule, descending limb of the loop, remember, permeable to water. Here we see our ascending limb, sodium potassium pumps. So we're pumping out sodium, chlorides following. Here we get into our distal limb and now the collecting tubule. And what I want us to focus on are the urea arrows. Here are the urea transporters. And this is going to diffuse and help create that hypertonic environment. But when we get to the ascending limb, you can see we're going to reabsorb that back into the ultrafiltrate. And as that gets absorbed back into the ultrafiltrate, guess what? It's going to recirculate around. We get to the collecting tubule, out it comes. Back in, do you see the cycle that's going to be created here? Urea is going to assist in maintaining that hypertonic environment here in the medullary region. Now the collecting tubule, this is going to be the last place where we have a chance to modify our ultrafiltrate, and it does a great job of this. In the collecting tubule, we're also going to have a means with which we can more rapidly reabsorb water, and that's going to be through those aquaporins that we talked about. And those aquaporins are going to be produced in response to anti-diuretic hormone, or ADH. We also know it as vasopressin. Now, that collecting tubule, it's going to run through the medulla of the kidney. It's going to run through this hypertonic environment. So this is where we create that hypertonic environment. We're finally going to adjust that last 27 liters that we had to play with. A lot of that's going to be either reabsorbed or more of that kept in the filtrate based on our response in the production of aquaporins to anti-diuretic hormone. So again, aquaporins are protein channels that have a hollow hydrophilic center to them. And as ADH increases, as you produce more ADH, you're going to increase the number of aquaporins and therefore have more water reabsorbed back into the kidney. But even when we get in periods of dehydration, we're going to see that anti-diuretic hormone cannot lead to more concentrated urine above that of the interstitial fluid. We're just going to get to the point that we have that obligatory water loss, and we just can't produce any more. We can't reabsorb any more than that. And it's especially during those extreme times of dehydration. Here we see an example of an aquaporin. We've got our phospholipid bilayer in the cells of the collecting tubule. Here we can see just a ribbon diagram of a protein. We can see some of these molecules, these charged molecules, creating this hydrophilic, basically, corridor that allows water molecules simply to diffuse via osmosis in the direction of their osmotic gradient. So here we see response to ADH. When we have dehydration, we're going to get those osmoreceptors in the hypothalamus. We talked about those in regulating the blood volume. The posterior pituitary is going to produce ADH in increasing amounts. The kidneys are going to reabsorb more water, and you're going to excrete more urine. And it's going to be a more concentrated urine. That's why they painted this a darker yellow. When we have too much water, we're overhydrated. This is going to have the opposite effect. ADH decreases. We decrease the number of aquaporins. Water reabsorption goes down, and now we produce a greater volume and a more diluted form of our urine. So as we end our second podcast, let's watch this video that is going to bring together not only the countercurrent multiplier, but the association with the vasorectae, and finally, this last absorptive function of the collecting tubules.
The loop of Henle creates a hyperosmotic region in the medulla of the kidney, acting as a sponge, drawing water out from the collecting duct, if the duct becomes permeable under the influence of vasopressin, also known as antidiuretic hormone, or ADH. The ascending limb of the loop of Henle, which is impermeable to water, pumps out sodium chloride via sodium-potassium chloride cotransporters from the urine into the blood in the vasa recta. Blood in the vasa recta carries this sodium chloride into the medulla, making the entire region hyperosmotic. The blood, which is moving in the opposite direction to the urine in the loop of Henle, then ascends parallel to the descending limb of the loop of Henle. This descending limb is water permeable, allowing water to move out of it into the vasa recta. This entry of water into the ascending vasa recta reduces the osmotic potential of the blood to normal as it returns to the body. The interplay between the loop of Henle and the vasa recta is called the countercurrent multiplier. Essentially, the medulla acts as a sponge, retaining and releasing water to address the water requirements of the body. ADH acts on the collecting duct to make it permeable to water, allowing water to move into the hyperosmotic medulla, where it is drawn up by the vasa recta and returned to the body.